Some animals can regrow what they lose. A salamander can rebuild a limb. Many creatures close wounds with a speed that feels almost effortless. Humans, in contrast, can survive decades, invent hospitals and map the genome. Yet a cut on our skin still needs time, still risks infection, still leaves a scar. The cold truth is that our bodies are not designed for perfect repair. They are designed for good enough, as long as we stay alive long enough to reproduce. In evolution, healing is not a promise. It is a compromise. What, exactly, did we trade away to become human? When we look at the natural world, it is easy to assume that healing should be perfect. A broken branch can grow back. A cut on the skin can close. Some animals even replace what they lose. From a distance, life seems built to repair itself. Yet for humans, healing often feels slow, incomplete, and marked by scars. A small wound can take days to close. A broken bone can take months. A deep injury may never return to its original state. To understand why, we have to step back to the beginning of our story. Long before hospitals or medicine, long before cities, when our ancestors lived in small groups on open land and forest edges, evolution does not work with goals or promises. It does not plan for comfort. It does not aim for perfection. It simply favors traits that help a species survive long enough to pass on genes. For early humans and their primate ancestors, survival meant finding food, avoiding predators, and raising children. If a wound closed well enough to prevent death from blood loss or infection, that was often sufficient. There was no need for skin to return to its exact original pattern. There was no reward in evolution for a scar-free body if the individual could still walk, hunt, gather, and reproduce. In the environments where early Homo species lived, energy was limited. Food had to be gathered, hunted, or scavenged. There were no steady supplies. Every calorie mattered. Healing requires energy. The body must stop bleeding, send immune cells to clean damaged tissue, build new blood vessels, and create new cells to close the wound. All of this costs fuel. For a species already investing heavily in a growing brain, which consumes a large portion of daily energy, there was little room for waste. The human brain, even in early Homo erectus, demanded more energy than the brains of most other mammals of similar size. That energy had to come from somewhere. In this setting, a slow but steady repair process made sense. The body closes a wound quickly at the surface to prevent infection. Beneath that surface, it rebuilds tissue in a way that is strong, even if not identical to what was there before. Scar tissue is not a failure. It is a fast solution. It trades perfect structure for stability. In a world where a small cut could expose someone to soil bacteria or animal bites, speed of closure mattered more than cosmetic precision. Evolution favored bodies that could seal damage reliably, even if the result was stiff or visible. When we compare ourselves to animals that appear to heal faster, we must remember that many of them follow different life strategies. Some small animals live only a few years. Their bodies grow quickly, reproduce quickly, and repair quickly. But they also age quickly and die young. Humans took another path. As our ancestors formed cooperative groups, shared food, and protected one another, survival did not depend solely on individual physical strength. A person with a healing injury could be supported by others. This social buffer reduced the pressure to evolve extremely rapid regeneration. In archaeological sites, we find skeletons of individuals who survived serious injuries, including broken limbs that healed over time. These remains show that even with slow healing, early humans could live on, supported by their community. So the question begins to shift. Healing is not slow because the body is careless. It is slow because it is balanced. Balanced between energy use and safety, between growth and control, between speed and stability. From the very beginning of our lineage, our bodies were shaped not for flawless repair, but for reliable survival in small, interdependent groups. The marks on our skin, the months it takes for bone to knit, the scars that remain, these are not signs of weakness. They are signs of an ancient compromise, written into us long before modern life changed the world around us. If evolution does not promise perfect healing, then we must ask what it truly protects. The answer becomes clearer when we imagine the daily life of early humans. They walked long distances. They climbed, carried, lifted, and ran. They lived close to the ground, close to soil, insects, and sharp stone. Injury was not rare. It was ordinary. A cut from stone tools, a fall while hunting, a bite during conflict, or a puncture from a thorn could all break the skin. In such a world, the first danger was not how the wound would look later. The first danger was blood loss and infection in the hours that followed. The human body responds to injury in stages. First it stops bleeding. Blood vessels tighten. Platelets gather. A clot forms. This happens quickly, often within minutes. Then comes inflammation. The area becomes warm, red, and swollen. Immune cells arrive to destroy bacteria and clear damaged tissue. This stage may feel uncomfortable, but it is protective. Only after these urgent tasks does the slower rebuilding begin. New tissue forms. Collagen is laid down. Over time, the wound contracts and closes. Each step follows a sequence shaped by millions of years of survival in unpredictable environments. For our ancestors in Africa, where the genus Homo first evolved, the environment was not sterile. Microbes were everywhere, in soil, in water, on animal skin. A wound that remained open too long increased the chance of deadly infection. In that context, the body's priority becomes understandable. It is better to close quickly and create a strong barrier, even if the structure is not identical to the original tissue. Scar tissue is rich in collagen fibers arranged in a simple, dense pattern. It lacks some of the flexibility and features of normal skin, such as hair follicles or sweat glands. Yet, it is durable. It protects what lies beneath. This approach reflects a deeper pattern in human biology. Our species invests heavily in stability. The human brain is complex and delicate. Our immune system must distinguish between harmful invaders and our own cells. Our bodies maintain tight control over cell division. Rapid, uncontrolled growth can lead to tumors. When tissue regenerates too aggressively, the risk of cancer rises. From an evolutionary perspective, there is constant pressure to balance repair with control. Too little repair, and injuries become fatal. Too much uncontrolled growth, and cells may divide in dangerous ways. If we look at animals that regenerate more dramatically, such as certain amphibians, we see a different balance. Some salamanders can regrow limbs. Their cells near the injury site can revert to a more flexible state, then multiply and form new structures. Humans do not do this. Our cells are more restricted in what they can become once they are specialized. This restriction protects the body from chaotic growth. It preserves organization, especially in complex organs. In the long arc of our evolution, as brains expanded and bodies became more intricate, tighter control likely offered more advantage than open-ended regeneration. There is also a social dimension to consider. Early humans did not heal alone. Archaeological findings show individuals with severe injuries who survived for years. A healed femur, for example, suggests that someone who could not walk for months was supported by others. This changes the evolutionary equation. If a group helps injured members survive, there is less pressure for extreme biological regeneration. Healing can afford to be slower if care and cooperation fill the gap. In this light, slow healing is not a defect. It is part of a larger system, a system that values controlled growth, infection defense, and long-term stability over rapid reconstruction. From the first Homo species walking across open savannas to later communities building shelters and sharing food, survival depended not on perfect tissue repair, but on balance within the body and within the group. When we imagine healing, we often imagine improvement. We imagine the body correcting damage and returning to its original state. Yet from the perspective of deep time, the body is not trying to restore perfection. It is trying to avoid catastrophe. For early humans, catastrophe did not mean a visible scar. It meant blood loss, infection, weakness, and death before raising children. The difference between life and death often unfolded within days, not years. In that narrow window, speed and control mattered more than refinement. If we return to the landscapes where our ancestors lived, open grasslands, woodland edges, riverbanks, we can begin to see why. These environments were physically demanding, movement was constant, food was uncertain. A serious injury reduced mobility, which reduced access to food and safety. A wound that stayed open invited bacteria. The body's solution was practical: seal the wound, stop the bleeding, activate inflammation to fight microbes, then rebuild enough structure to bear weight and resume movement. The goal was functional recovery, not architectural perfection. Over generations, natural selection preserved this strategy. Individuals whose bodies responded too slowly to bleeding or infection were less likely to survive long enough to reproduce. Individuals whose cells grew too freely faced another risk: uncontrolled growth, which can damage organs and shorten life. The human lineage gradually leaned toward regulation. Cell division became tightly monitored. Specialized tissues remained specialized. Once a skin cell becomes a skin cell, it rarely reverts to a more flexible state. This limits the ability to regenerate entire structures, but it increases stability across a long lifespan. The length of that lifespan is important. Humans do not mature quickly and die young like many small mammals. Even in prehistoric times, although average life expectancy at birth was low due to infant mortality, many adults who survived childhood could live into their 40s or beyond. This extended adulthood created a different biological pressure. A body that must function for decades benefits from careful maintenance and strict growth control. Rapid regeneration systems, which rely on highly flexible cells, may increase the risk of mutations over time. In a long-lived species, accumulated mutations carry greater consequences. Archaeological evidence quietly supports this picture. Skeletons from Paleolithic sites show healed fractures, fused vertebrae, and signs of past trauma. These individuals did not regenerate perfectly. Their bones healed with irregular shapes, joints stiffened, yet they survived long enough for healing to complete. This suggests that early human communities tolerated imperfection in exchange for survival. A person with a healed but altered limb could still contribute knowledge, tool-making skills, or social bonds. The body did not need to restore every detail to maintain the continuity of the group. This broader view reframes the original question: why do humans heal slowly compared to other animals? Because our species did not evolve in isolation from time, energy, and community. Our biology reflects compromise. It reflects limited calories in ancient environments, the high cost of a growing brain, the dangers of infection, and the risks of uncontrolled cell growth. It reflects a social structure in which injured individuals were not always abandoned, reducing the pressure for extreme biological repair. From the earliest members of the genus Homo to modern humans, the pattern remains consistent. We close wounds reliably, we rebuild bone carefully, we form scars that mark where damage once occurred. These marks are reminders that healing was never about erasing the past, it was about continuing into the future. The body carries forward, even if altered. That quiet persistence, steady, regulated, and sometimes slow, was enough to allow our ancestors to survive long enough to become us. Healing may look like a simple act from the outside, but inside the body it is an expensive process. Every repair requires energy. When skin breaks, when muscle tears, when bone cracks, the body must mobilize fuel to stop bleeding, activate immune cells, build new tissue, and reshape damaged areas. None of this happens without cost. For early humans living in unpredictable environments, energy was not stored in abundance. It had to be gathered daily, often with effort and risk. In that context, healing could never be unlimited or wasteful. It had to compete with other demands. The human brain alone consumes a remarkable share of daily energy. Even at rest, it uses a large portion of the calories we take in. This was already true in our ancient ancestors. As brain size increased over the course of human evolution, from earlier Homo species to Homo sapiens, the energetic burden grew. A larger brain supported memory, planning, cooperation, and tool use. These traits improved survival, but they also required steady fuel. When food was scarce, the body had to make decisions. Should energy go to repairing a minor injury at full speed, or to maintaining brain function, body temperature, and immune defense? Inside the body, energy is distributed carefully. Cells rely on molecules such as glucose and fatty acids to power their work. When tissue is damaged, specialized cells begin to multiply and produce new material, especially collagen, the protein that gives structure to skin, tendons, and bone. Collagen synthesis is not cheap. It demands amino acids, oxygen, and metabolic effort. At the same time, immune cells consume energy as they engulf bacteria and clear debris. Blood vessels expand and grow new branches to nourish the area. All of this increases metabolic demand. In the world of early humans, long periods of reliable surplus were rare. Hunting could fail. Seasons could shift. Drought could reduce plant availability. In such conditions, a system that required massive energy for perfect regeneration would be risky. A body that insists on rebuilding every detail before allowing movement might fall behind in a competitive environment. Instead, human healing follows a measured pace. It prioritizes sealing and stabilizing first. It allows gradual strengthening later. This approach spreads the energy cost over time. If we compare this with animals that regenerate more dramatically, we see different life strategies. Some amphibians that regrow limbs often live in environments where metabolic rates and life histories differ significantly from ours. Their bodies are organized in ways that allow certain cells to return to a more flexible state after injury. Yet these systems function within species that do not carry the same energetic investment in a large brain or long childhood. Human children require years of care before reaching independence. Supporting this long developmental period already demands significant resources from adults and from the group as a whole. The energetic trade-offs are not abstract ideas. They shape real biological limits. When people experience severe malnutrition, wound healing slows further. This is well documented in medical observation. Without adequate protein, vitamins, and calories, the body struggles to rebuild tissue. This modern evidence echoes ancient realities. Our ancestors' bodies were tuned to environments where resources fluctuated. Healing had to function within those limits. Seen this way, slow healing is not laziness or failure. It reflects a careful accounting system written into our metabolism. The body does not repair at maximum speed because maximum speed is rarely sustainable. Instead, it balances the cost of repair with the need to keep the organism functioning day after day. From the earliest humans walking across open landscapes to us today, this balance has remained. Energy must be shared, and healing must wait its turn. Energy inside the human body is not simply available for any one purpose. It is constantly divided, redirected, and prioritized. When an injury occurs, the body does not suddenly gain extra fuel. It must draw from the same limited pool that supports breathing, circulation, temperature regulation, digestion, brain activity, and movement. In our modern lives, with regular meals and climate control, it is easy to forget how narrow those margins once were. For most of human history, the body operated close to its limits. In the early environments of our species, energy intake varied from day to day. A successful hunt could provide abundance. A failed hunt could mean scarcity. Gathering wild plants depended on season and rainfall. Under these conditions, the body evolved systems that were cautious. It stored energy as fat when possible, but it did not assume constant surplus. When injury happened, the repair process had to fit into this unpredictable landscape. Healing is not a single action. It unfolds in phases, each demanding different resources. After clotting and inflammation, the body begins rebuilding tissue. Fibroblast cells produce collagen. New blood vessels form to supply oxygen and nutrients. Later, tissue is reorganized and strengthened. Each of these stages requires raw materials: proteins for structure, lipids for cell membranes, carbohydrates for immediate fuel, and micronutrients such as vitamin C and zinc for specific biochemical steps. Without these inputs, healing slows. This is not because the body lacks intelligence. It is because biology obeys physical limits. When energy is scarce, the body protects what is most essential in the short term. The brain, heart, and lungs cannot pause. Core temperature must be maintained. Muscles must still function well enough to find food or avoid danger. If healing were to consume too much energy at once, it could weaken these critical systems. A measured pace spreads the cost. It allows the organism to remain active while repair proceeds gradually. There is also a deeper evolutionary trade-off at work. Rapid growth and rapid repair both depend on increased cell division, yet increased cell division carries risk. Every time a cell copies its DNA, there is a chance of error. Over a long lifespan, these errors accumulate. In a short-lived animal, the window for dangerous mutations is brief. In humans, who can live for many decades, strict control over cell division becomes more valuable. A system that limits uncontrolled growth may heal more slowly, but it reduces the likelihood of harmful mutations developing into cancer. Our long childhood adds another layer to this balance. Human offspring depend on adults for many years. This prolonged dependency means that adults must remain functional over extended periods. If adult bodies were optimized only for rapid regeneration without strict regulation, the long-term risks might outweigh the short-term benefits. Evolution does not design for comfort. It designs for reproductive success across generations. A stable adult body capable of surviving moderate injuries over decades supports the survival of dependent children. Archaeological findings quietly echo this pattern. Skeletal remains from prehistoric sites show healed fractures and dental infections that resolved over time. The healing was not instant. Bones often fused imperfectly. Teeth show signs of long-term inflammation, yet individuals survived. This suggests that even with slow repair, the balance between energy use and stability was sufficient in the environments where humans evolved. The body's choice, if we can call it that, is conservative. It avoids extremes. It does not pour all resources into a single wound at the expense of the whole organism. Instead, it repairs steadily, within metabolic limits, while preserving long-term cellular control. In this steady rhythm, shaped by fluctuating food supplies and long lifespans, we can see why human healing rarely appears dramatic. It is deliberate. It is cautious. And it reflects an ancient economy of energy that still guides our cells today. If healing depends on energy, then the pace of healing is tied to the broader rhythm of life itself. In the ancient world where humans evolved, that rhythm was rarely smooth. Food supplies rose and fell with seasons. Climate shifted over generations. Groups migrated in response to drought, cold, or the movement of herds. Under these changing conditions, the human body had to remain adaptable, not just strong. Energy could not be spent carelessly, because tomorrow's supply was never guaranteed. When a wound occurred in such an environment, the body responded within these limits. It sealed, protected, and repaired in stages that matched available resources. During times of abundance, healing may have progressed more quickly. During scarcity, it likely slowed. This flexibility was not visible from the outside, but it was built into our metabolism. Hormones such as cortisol and insulin shift according to stress and food intake, influencing how nutrients are stored and used. These same shifts affect immune activity and tissue repair. The body does not treat healing as separate from survival. It integrates it into the same system that governs hunger, stress, and growth. Even today, modern medical observation shows how tightly healing is linked to nutrition and metabolic state. People with severe malnutrition, chronic illness, or uncontrolled diabetes often experience delayed wound repair. High blood sugar can interfere with immune response and collagen formation. Low protein intake limits the raw materials needed for rebuilding tissue. These clinical patterns reflect ancient constraints. Our bodies still operate on the assumption that resources may be limited. When internal balance is disturbed, repair slows because the system prioritizes stability over speed. This connection between metabolism and healing becomes clearer when we consider the long arc of human evolution. As our ancestors' brains expanded, energy demands increased. A large brain offered advantages in memory, tool use, and cooperation, yet it required consistent fuel. Compared to other mammals of similar size, humans allocate a greater share of daily energy to brain function. This allocation leaves less room for extravagant biological processes elsewhere. Healing therefore proceeds in a way that conserves resources, even if it means accepting visible scars or longer recovery times. There is also the matter of mobility. Early humans depended on movement for survival. If healing required total immobility for extended periods, individuals would have struggled to obtain food or remain safe. A slower but functional recovery allowed continued participation in group life. A person with a healing fracture might move carefully, supported by others, rather than being entirely incapacitated by an aggressive regeneration process, demanding total rest and high energy input. In this way, moderate repair aligned with the demands of an active, mobile species. Across archaeological sites, we find evidence of this balance. healed fractures, worn joints, and signs of past infections reveal that injuries were common but survivable. These remains suggest that healing was sufficient to restore function, even if it did not restore perfection. The skeleton records adaptation rather than ideal symmetry. It shows that the body's aim was continuity, not flawlessness. When we step back, the pattern becomes steady and clear. Human healing is shaped by an ancient economy of energy. It reflects fluctuating food supplies, the high cost of brain function, long developmental periods, and extended lifespans. The body repairs within limits because those limits once protected survival. What appears slow today is part of a strategy that has endured for hundreds of thousands of years. Our cells still follow the same quiet accounting system, balancing every act of repair against the deeper need to keep the whole organism alive over time. When a wound closes on human skin, it rarely disappears without a trace. A thin, pale line may remain. Sometimes the mark is thicker, raised, or slightly darker than the surrounding tissue. We call it a scar. It is easy to think of a scar as evidence that healing failed to restore what was lost. Yet, from a biological perspective, a scar is proof that the body succeeded in its first priority, sealing damage quickly enough to prevent something worse. To understand why scars exist, we must look closely at what happens after the initial stages of clotting and inflammation. Once bleeding is controlled and immune cells have reduced the immediate threat of infection, the body begins building new tissue. Specialized cells called fibroblasts migrate into the wound. They produce collagen, a strong structural protein that forms the framework of skin and connective tissue. In normal skin, collagen fibers are arranged in an organized pattern that allows flexibility and resilience. In scar tissue, the fibers are laid down more rapidly and in a denser, simpler arrangement. The goal is strength and closure, not fine detail. This difference is not accidental. In the environments where early humans evolved, open wounds were dangerous. Soil, plant matter, and animal contact exposed broken skin to bacteria. There were no antiseptics, no antibiotics. A wound that remained open too long increased the chance of infection spreading into deeper tissues or the bloodstream. Under these conditions, the body's strategy favored speed. Rapid deposition of collagen created a barrier. Even if the new tissue lacked hair follicles or sweat glands, it reduced the immediate risk. The presence of scars in ancient skeletal remains supports this understanding. Archaeologists have documented healed fractures and evidence of soft tissue injury inferred from bone remodeling. These healed areas often show irregular shapes or thickened bone where repair occurred. The body did not recreate the exact original structure. Instead, it reinforced the area enough to restore basic function. In the context of prehistoric life, this was sufficient. A hunter or gatherer did not need perfect symmetry. They needed strength to walk, carry, and protect themselves. There is also a deeper biological reason for the formation of scars. Perfect regeneration would require cells at the injury site to revert to a more flexible state, capable of recreating complex structures. In humans, most adult cells are specialized and tightly regulated. They rarely return to a less differentiated form. This restriction reduces the risk of uncontrolled growth. Scar formation is part of this regulatory framework. By rapidly stabilizing tissue with dense collagen, the body avoids prolonged periods of active cell division that might increase the chance of genetic errors. Scars then reflect a careful compromise between repair and control. They close wounds quickly, reduce exposure to pathogens, and restore mechanical strength. They do not aim to replicate the original blueprint in detail. In a species that evolved to live for decades and invest heavily in brain development, strict regulation of cell growth may have offered greater long-term advantage than the ability to regenerate perfectly. If we imagine an early human with a deep cut from stone or bone, the body's priorities become clearer. Bleeding must stop. Infection must be contained. The wound must close before the next day's journey. A visible scar is a small price compared to survival. From the first members of our lineage walking across African landscapes to modern humans today, this pattern remains. Our skin remembers injury not because it failed, but because it chose speed and stability over flawless restoration. If scars are the visible result of rapid closure, then it becomes important to ask what would be required for something different. To restore skin perfectly, the body would need to recreate not only the surface layer, but also the intricate structures beneath it: hair follicles, sweat glands, pigment cells, and networks of nerves. Each of these elements develops through carefully timed signals during early growth. In adults, those developmental pathways are largely quiet. They are not easily reactivated. In the womb and during early childhood, human tissues possess greater flexibility. Cells divide more readily. Growth factors circulate in higher amounts. In fetal development, wounds can sometimes heal with minimal scarring. This observation has been documented in medical research and provides an important clue. The young body is still in a state of active growth. Its cells are less restricted and more capable of rebuilding organized structures. As development progresses, however, the system shifts towards stability. Cells become specialized. Gene expression patterns become fixed. The emphasis moves from building new organs to maintaining what already exists. This shift is not accidental. As humans evolved larger brains and longer lifespans, the cost of cellular instability increased. Each time a cell divides, it must copy its DNA. Errors can occur. Most are repaired, but some persist. Over decades, accumulated errors can lead to malfunction or cancer. A system that allows widespread cellular reprogramming in adulthood might enhance regeneration, but it would also raise long-term risk. In a species that often survives well past reproductive age and invests heavily in offspring, minimizing such risk carries evolutionary weight. Looking at archaeological evidence again, we see bones that healed with thickened callus formations where fractures occurred. The callus is essentially scar tissue in bone form. It bridges the broken ends and restores strength. It does not recreate the original smooth contour immediately. Over time, some remodeling occurs, but traces remain. These skeletal records reveal that human repair mechanisms have long favored reinforcement over exact restoration. The bone becomes strong enough to bear weight and withstand stress, even if its shape differs slightly from before. Environmental pressures likely reinforced this strategy. Early humans faced frequent minor injuries from tools, travel, and close interaction with animals. A system designed for perfect regeneration might require prolonged immobility and high energy input. In contrast, scar-based repair allows individuals to resume activity sooner, even if not at full strength. Function returns gradually. This gradual return fits the demands of a mobile, cooperative species. There is also a sensory dimension. Scar tissue contains fewer specialized structures, including some nerve endings. In certain cases, this may reduce sensitivity in the healed area. While this can limit fine perception, it may also reduce persistent pain signals from previously injured tissue. The trade-off is subtle but reflects the broader theme of compromise rather than perfection. When we consider all these factors together, developmental pathways, DNA stability, archaeological patterns, and environmental demands, the presence of scars becomes less mysterious. They are not signs that human biology is inferior to that of other animals. They are markers of a system optimized for durability across long spans of time. Our ancestors survived not because their bodies erased every injury, but because they stabilized damage efficiently and continued forward. The faint lines on human skin echo that ancient decision, shaped long before written history, when survival depended on balance rather than flawless repair. If we follow the story of scars to its quiet conclusion, we begin to see that they are not simply marks of damage. They are records of how the human body chooses to solve a problem. Faced with injury, the body does not attempt to rewind time. It does not search for the original blueprint and rebuild every detail. Instead, it stabilizes, it strengthens, it restores enough structure to continue living. The trace that remains is not a mistake. It is the visible edge of a deeper strategy. To understand this fully, we can return once more to the earliest environments of our species. In the shifting climates of the African Pleistocene, early humans moved through open savannas, woodland patches, and river valleys. They carried tools made of stone and bone. They hunted, butchered, and gathered. Their lives involved friction between skin and rough surfaces, between muscle and weight, between bone and sudden force. Injury was part of movement. Under these conditions, the body's priority was not aesthetic restoration. It was structural resilience. Scar tissue, though less flexible, is strong. It binds tissue quickly. In bone, a thick callus forms around a fracture site, temporarily enlarging the area. Over months and years, the bone remodels, but the process remains conservative. The body does not erase the memory of the break, it integrates it. In skin, collagen fibers align in dense bundles that resist tearing. The healed area may stretch less, but it protects against reopening. For a species constantly exposed to environmental stress, this reinforcement could mean the difference between continued mobility and repeated injury. There is also a generational perspective. Humans developed long childhoods and extended social bonds. Survival was no longer only about individual strength. Groups cared for injured members. Archaeological evidence shows individuals with severe injuries, damaged limbs, degenerative joint disease who survived for years. Their scars, internal and external, became part of their history. This social support reduced the evolutionary pressure for flawless biological repair. If the group could compensate for physical limitation, the body did not need to regenerate perfectly to ensure survival and reproduction. At the cellular level, scar formation reflects strict regulation. Once a wound is closed, growth signals diminish. Fibroblasts reduce activity. Collagen deposition slows. This controlled shutdown prevents runaway cell division. It may leave behind a visible mark, but it also protects long-term stability. In a species that can live for decades, this restraint becomes critical. Perfect regeneration might require prolonged periods of intense cell proliferation. Over time, such activity could increase the chance of harmful mutations. Seen across deep time, scars represent a compromise between flexibility and safety, between speed and precision. They show that the human body values closure over recreation. They also remind us that healing is not a return to an untouched state. It is a forward movement, integrating injury into the body's ongoing story. When we ask why humans heal slowly compared to other animals, scars provide part of the answer. Our repair system is designed to contain risk, conserve energy, and preserve long-term stability. It may not restore every detail, but it reliably restores function. In the lives of our ancestors, that reliability was enough. The faint lines on our skin today are echoes of that ancient logic, a logic that chose endurance over perfection and continuity over flawless renewal. When we compare humans with many other animals, one quiet difference stands out. Humans often live longer, even in prehistoric times. Although many children did not survive infancy, adults who reached maturity could live for decades. Some skeletal remains suggest individuals surviving into their 40s, 50s, and beyond. In evolutionary terms, that is a long time. A longer lifespan changes the biological equation. It reshapes how the body manages growth, repair, and risk. In species that live fast and die young, the priorities are different. Small mammals, for example, often grow quickly, reproduce early, and experience high turnover. Their bodies may tolerate faster cell division and rapid tissue repair because the window of life is short. There is less time for accumulated cellular errors to cause serious harm. In such species, regeneration and rapid healing can fit within their life strategy. The emphasis is on speed. Humans followed another path. As brain size expanded and social complexity increased, our species developed extended childhood and prolonged adulthood. Children required years of care before they could survive independently. Adults, in turn, needed to remain functional long enough to raise offspring and contribute to the group. This extended lifespan created a new pressure, stability across time. A body that must endure decades of use must also guard against long-term threats, including uncontrolled cell growth. Every act of healing involves cell division. New skin cells, new bone cells, new blood vessels must form. Yet each division carries a small risk of mutation. In a short-lived animal, these risks may rarely accumulate to dangerous levels before death from other causes. In humans, who can live much longer, the cumulative risk increases. Evolution therefore favored stricter control over when and how cells divide. This control protects against cancer but also limits dramatic regeneration. The fossil record offers quiet support for this idea. Compared to many animals, humans show patterns of age-related wear in bones and joints that reflect long-term survival. Signs of arthritis, healed injuries, and gradual degeneration appear in ancient remains. These individuals did not regenerate damaged tissue perfectly. Instead, they adapted to change and continued living. The body prioritized containment of damage over full restoration. There is also a demographic dimension. In small prehistoric groups, older individuals carried knowledge about migration routes, edible plants, tool-making, and social bonds. Even if their physical strength declined, their presence could benefit the group. This may have reinforced selection for longevity and for systems that reduce catastrophic failure over time. Slower, regulated healing fits within this framework. It avoids aggressive cellular behavior that might threaten long-term survival. Long life demands careful balance. The body must repair enough to remain functional, but not so aggressively that it destabilizes itself. In this sense, slow healing is intertwined with longevity. The same regulatory systems that reduce cancer risk and maintain tissue organization over decades also restrain rapid regeneration. What may seem like a weakness in comparison to certain animals is, in fact, part of a broader strategy aligned with extended human life. From early members of the genus Homo to modern populations, this pattern persists. We heal steadily, not explosively. We scar, we remodel, we adapt. Over a lifespan that can stretch many decades, this conservative approach may offer greater security. In choosing stability over speed, the human body aligned itself with the long arc of its own existence. A long life does not simply extend the calendar, it changes the mathematics of risk. In a body that may function for 40, 50 or more years, every biological decision must account for time. A small error, repeated over decades, can grow into something serious. For this reason, the human body tends to favor caution in processes that involve growth and repair. Healing is not only about closing a wound today, it is also about protecting the body 10 or 20 years from now. Cell division lies at the center of this balance. To repair tissue, cells must multiply. Each time a cell divides, its DNA is copied. The copying process is remarkably accurate, supported by repair enzymes that correct mistakes. Yet no system is perfect. Tiny errors can occur. Most are harmless, some are repaired, a few may alter how a cell behaves. In a short-lived animal, the chances that such changes will accumulate to dangerous levels before death from other causes are relatively small. In humans, who often live well past reproductive age, the window of accumulation is longer. This extended window likely shaped how human tissues regulate regeneration. Cells are generally more restricted in what they can become. Growth signals are tightly controlled. Tumor suppressor genes monitor division and can trigger cell death if abnormalities are detected. These protective systems reduce the likelihood of uncontrolled growth, but they also slow the pace of repair. Regeneration that is dramatic and flexible may carry a higher risk in a species built for longevity. If we look again at skeletal remains from ancient populations, we see evidence of gradual adaptation rather than rapid renewal. Bones show remodeling after fractures, but the process unfolds over months and years. Joints display wear that reflects decades of use. Teeth reveal layers of growth that record seasonal stress. These signs tell us that the human body is organized around endurance. It adjusts slowly, reinforces slowly, and maintains structure across long spans of time. There is also a generational layer to this story. Humans evolved extended family systems. Grandparents may have contributed knowledge and care that improved survival of younger members. This idea, sometimes called the grandmother hypothesis, suggests that longevity itself offered evolutionary advantage. If older individuals improved the survival of grandchildren, genes supporting longer life would be favored. Within this framework, the body must be reliable across decades, not optimized for rapid short-term regeneration. Rapid, flexible cell growth might help regrow tissue more perfectly after injury. Yet such flexibility could undermine stability over time. A long-lived organism benefits from predictability in cell behavior. Controlled repair, even if slower, reduces uncertainty. It preserves the integrity of complex organs, especially the brain, whose function depends on stable networks of cells that are not easily replaced. Seen from this wider perspective, slow healing becomes part of a longevity package. The same biological safeguards that allow humans to live long enough to raise children and support grandchildren also limit the speed of regeneration. The body trades dramatic repair for durability. It accepts scars and gradual recovery in exchange for long-term order. Over thousands of generations, this cautious strategy may have offered greater overall success than the faster but riskier alternatives seen in shorter-lived species. If a long life changes the balance of risk, it also changes the meaning of recovery. In a short-lived animal, healing must serve a brief future. In humans, healing supports a future that may stretch across decades. The body does not repair only for tomorrow's survival, but for years of movement, work, and social connection. This extended horizon influences how repair unfolds. Consider bone healing once more. When a fracture occurs, the body forms a temporary bridge of tissue between the broken ends. This bridge gradually hardens into a callus. Over time, the bone reshapes itself along lines of stress, a process known as remodeling. This remodeling can continue for months or even years. The pace may seem slow, yet it produces a structure capable of bearing weight repeatedly over long spans of life. In a species that expects decades of use, this careful strengthening makes sense. The same pattern appears in skin and muscle. After injury, tissue regains strength gradually. Collagen fibers align along directions of tension as the person resumes activity. This alignment improves durability. It is not immediate. It is shaped by daily movement. In early human environments, where walking long distances and carrying loads were common, tissues needed to adapt to repeated stress. Rapid regeneration without proper alignment could create weakness. Slower remodeling allowed integration with the body's ongoing patterns of use. Longevity also magnifies the consequences of chronic conditions. Inflammation, for example, is protective in the short term, but harmful if persistent. A long-lived organism must regulate inflammatory responses carefully. Excessive, prolonged inflammation can damage tissues and accelerate aging. Therefore, the human immune system includes feedback mechanisms that gradually reduce inflammatory signals once immediate threats are controlled. This moderation may extend healing time, yet it protects long-term tissue integrity. Demographic evidence from ancient populations suggests that individuals who survived childhood often contributed to group stability for many years. Even with healed injuries or physical limitations, older adults could provide knowledge, childcare, and coordination. A body designed for extended participation benefits from cautious repair. It prioritizes maintaining overall balance rather than maximizing speed in any single event. The trade-offs of longevity are visible even in modern medicine. Treatments that stimulate rapid cell growth can aid recovery in some contexts, but they must be used carefully because of potential long-term risks. Similarly, therapies that suppress inflammation may reduce pain but can interfere with necessary immune processes. These medical realities mirror ancient biological compromises. The human body has evolved to weigh immediate repair against future stability. When we step back across evolutionary time, the pattern remains consistent. Humans did not evolve as brief, rapidly cycling organisms. We evolved as long-lived, socially interdependent beings. Our healing reflects this identity. It unfolds steadily, controlled by safeguards that reduce risk across decades. What appears slow when compared to certain animals is, in fact, aligned with a life strategy built around endurance and continuity. Slow healing, then, is not separate from longevity. It is woven into it. The same regulatory systems that allow humans to survive into later adulthood also restrain dramatic regeneration. The body chooses careful rebuilding over rapid replacement because it anticipates a long journey ahead. In this light, the pace of human healing becomes part of a larger design shaped by time itself. When the skin breaks or tissue is damaged, the body does not respond quietly. One of the first reactions is inflammation. The area becomes red, warm, swollen, sometimes painful. These signs can feel like a problem, yet they are part of a protective process that has been shaped over millions of years. Inflammation is not an accident of biology. It is one of the oldest defense systems shared across many forms of life. To understand why humans heal as we do, we must understand why inflammation exists at all. In the environments where early humans lived, microbes were constant companions. Soil, water, animal hides, and plant surfaces all carried bacteria. Without modern sanitation, a wound meant exposure to these organisms. Inflammation is the body's rapid alarm system. Blood vessels widen to increase flow to the area. Immune cells move from circulation into the damaged tissue. These cells engulf bacteria, remove debris, and release chemical signals that coordinate repair. The swelling that follows is a sign of increased fluid and cellular activity, not simply damage. This response was essential in prehistoric life. A small infection could become life-threatening if bacteria entered the bloodstream. Individuals whose immune systems reacted too slowly were less likely to survive. Natural selection therefore favored a strong and immediate inflammatory response. It was better to overreact slightly than to fail to contain an infection. In this sense, inflammation represents caution shaped by risk. Yet, inflammation is costly. It consumes energy and can damage nearby healthy tissue if prolonged. The chemicals released during the immune response increase metabolic demand. Fever, which sometimes accompanies infection, raises body temperature to slow microbial growth but also requires additional fuel. In an environment where food was not guaranteed, excessive or unnecessary inflammation could weaken the organism. The body therefore evolved feedback systems to limit the duration and intensity of this response. The balance is delicate. Too little inflammation leaves wounds vulnerable to infection. Too much prolongs healing and can harm surrounding structures. In humans, this balance is influenced by hormones, nutritional status, and stress levels. Cortisol, a hormone released during stress, can suppress certain aspects of immune function. In ancient settings, stress from danger or scarcity may have temporarily altered inflammatory responses. The body constantly integrates signals about safety, energy availability, and external threat when regulating repair. Archaeological evidence again provides subtle clues. Skeletal remains sometimes show signs of chronic inflammation, such as bone changes associated with long-term infection. These findings remind us that not every wound healed smoothly. Some injuries led to prolonged immune activity, yet many individuals survived these episodes, suggesting that, on average, the inflammatory system was effective enough to preserve life. In modern times, we often think of inflammation as something to reduce. Anti-inflammatory medications are common. However, the existence of these treatments should not obscure the fact that inflammation once protected our ancestors in environments far more dangerous than those we inhabit today. The redness and swelling around a cut are echoes of that ancient protective reflex. When we ask why humans heal slowly compared to other animals, inflammation offers part of the answer. It is powerful but controlled. It prioritizes containment of threat before reconstruction of tissue. It operates within energetic limits and under tight regulation to prevent long-term harm. This careful management can extend the visible course of healing, yet it reflects an ancient necessity. Survive the immediate danger first. Only then can rebuilding proceed. Inflammation begins as protection, but it does not automatically stop at the perfect moment. The same signals that summon immune cells and increase blood flow must also be silenced once the immediate threat is controlled. If they continue for too long, they can delay healing rather than support it. This tension between activation and restraint lies at the heart of human recovery. The body must know when to defend and when to rebuild. In the ancestral environments of our species, this balance was shaped by repeated exposure to injury and infection. A wound contaminated with soil or animal tissue required a strong inflammatory reaction. Immune cells released chemical messengers that attracted more defenders. These messengers also stimulated nearby cells to divide and repair tissue. Yet prolonged inflammation consumes resources and can damage surrounding healthy structures. Proteins and enzymes released to destroy bacteria may also break down the body's own tissues if not carefully regulated. For early humans living in small, mobile groups, excessive inflammation carried consequences. Fever, fatigue, and pain reduce mobility. A person who cannot move easily may struggle to gather food or keep pace with the group. Natural selection would therefore favor individuals whose inflammatory responses were strong enough to control infection, but flexible enough to taper off once the danger subsided. The immune system evolved layers of feedback signals to dampen its own activity, Specialized regulatory cells help calm the response after the peak has passed. This balance is not perfect. Even today, inflammation can persist longer than needed. Chronic inflammatory conditions, such as arthritis or certain autoimmune disorders, reveal what happens when regulation falters. These conditions remind us that the inflammatory system is powerful and complex. It evolved under pressures very different from those of modern life. In prehistoric contexts, most inflammatory episodes were tied to visible injuries or infections. In modern environments, where lifespan is extended and lifestyles differ, inflammatory responses may be triggered by factors such as metabolic imbalance or environmental stress. The archaeological record offers indirect insight into this ancient balance. Skeletal remains sometimes show evidence of healed infections in bone, suggesting that inflammation succeeded in containing bacteria but left structural changes behind. The body resolved the immediate threat, yet traces remained. These traces show that healing is not always a clean reversal. It is often a negotiation between damage control and tissue repair. Energy again plays a role. Sustained inflammation requires fuel. Immune cells rely heavily on glucose during active defense. In periods of scarcity, prolonged inflammatory responses could weaken the organism. Therefore, metabolic signals interact with immune pathways. When energy stores decline, the body may suppress certain immune functions to conserve resources. This interaction can influence the pace of healing, stretching it over a longer period while maintaining overall stability. Seen in this light, slow human healing is partly the visible result of this careful regulation. The body does not simply fight infection and then immediately regenerate perfectly. It moves through phases, each moderated by signals that weigh risk against resource availability. The inflammatory stage must rise quickly but also fall gradually. The rebuilding stage must proceed steadily without triggering uncontrolled growth. From the earliest Homo populations navigating unpredictable climates to modern humans managing complex lives, this regulatory dance has remained. Inflammation protects, but it must be contained. Healing advances, but it must respect limits. What appears slow from the outside reflects a system that constantly negotiates between defense and preservation, shaped by millions of years of balancing survival with long-term stability. Inflammation is not only a reaction to injury, it is also a memory of past environments. The human immune system was shaped in landscapes filled with microbes, parasites, and frequent minor wounds. For most of our evolutionary history, the greater danger was not excessive immune response, but insufficient defense. A cut that became infected could end a life. Under such pressure, natural selection favored vigilance. The inflammatory system became quick to activate and cautious to withdraw. Yet vigilance has consequences. When the immune system responds, it does not distinguish between inconvenience and catastrophe. The same chemical messengers that destroy bacteria can also break down surrounding tissue. Swelling can limit movement. Pain signals encourage rest. These reactions are useful in the short term. They reduce further damage and direct energy toward repair, but they also slow visible recovery. Tissue cannot rebuild fully until inflammation recedes. In early human communities, this temporary slowdown may have been tolerable. Cooperation provided a buffer. An injured individual could rest while others gathered food or protected the group. Archaeological discoveries of healed fractures and signs of long-term survival after injury suggest that community support was common. This social safety net allowed the inflammatory process to unfold at its natural pace without immediate catastrophic consequences. Healing did not have to be dramatic or instantaneous to ensure survival. However, the body must also guard against inflammation that persists too long. Prolonged immune activity can lead to tissue scarring, joint stiffness, or chronic pain. To prevent this, regulatory mechanisms evolved to dampen immune signals once pathogens are controlled. Specialized immune cells release anti-inflammatory factors. Hormones adjust the intensity of the response. This gradual tapering takes time. It reflects the body's attempt to avoid long-term damage while still ensuring immediate protection. The environment in which humans evolved required this careful moderation. Exposure to soil microbes, animal pathogens, and physical trauma meant that the immune system was frequently tested. At the same time, energy resources were limited, and life expectancy, though longer than many species, still required sustained bodily integrity over decades. A system that activated strongly but deactivated responsibly offered balance. It avoided fatal infection while reducing the chance of prolonged self-damage. Modern observations reveal the same pattern. When inflammation is suppressed too aggressively, infection risk rises. When it is left unchecked, chronic conditions develop. The body walks a narrow path between these extremes. Slow healing often reflects this negotiation. Tissue repair cannot proceed to completion until inflammatory signals decline to safe levels. The visible swelling, redness, and stiffness are signs that the immune system is working within its inherited framework. Seen across deep time, inflammation appears less as a flaw and more as a guardian with strict rules. It protects first, rebuilds second, and withdraws carefully. The pace may feel slow compared to animals with different life histories, yet it aligns with the human pattern of extended lifespan, social cooperation, and tightly regulated growth. Inflammation is both shield and constraint. It ensures survival in dangerous environments, even if it leaves behind traces of its intensity in the form of scars or prolonged recovery. In this way, the speed of human healing is inseparable from the character of our immune system. It is shaped by ancient dangers, limited energy, and the need for long-term stability. What seems like delay is often a sign of restraint, a restraint that once guarded our ancestors against threats far more immediate than those most of us face today. Among the many differences between humans and certain other animals, one stands out quietly but clearly. Some creatures can replace what they lose. Certain amphibians can regrow a limb. Some fish can regenerate parts of their heart. In contrast, humans cannot regrow an arm or a leg. When we lose complex structures, they do not return. This contrast often leads to a simple question. Did humans lose something during evolution? To approach this carefully, we must first understand that regeneration exists on a spectrum. Even in humans, regeneration is not absent. Skin renews itself constantly. The lining of the gut is replaced regularly. The liver can regrow significant portions of its mass after injury. Bone remodels over time. These examples show that our bodies retain regenerative capacity. Yet this capacity is limited when it comes to rebuilding entire structures with intricate organization, such as limbs or large segments of muscle and nerve. In animals capable of dramatic regeneration, cells near the injury site can revert to a more flexible state. They form a cluster of undifferentiated cells that multiply and later specialize again to recreate lost tissues. This process requires a level of cellular plasticity that is tightly restricted in adult humans. In our species, most mature cells remain committed to their specific roles. A skin cell remains a skin cell. A muscle cell remains a muscle cell. This specialization enhances stability, but reduces flexibility. Why would evolution favor restriction over flexibility? Part of the answer lies in complexity. As the human lineage evolved larger brains, intricate hands, and finely coordinated systems, maintaining organization became crucial. The human body contains highly specialized tissues arranged in precise patterns. The nervous system, in particular, depends on stable connections between billions of neurons. Allowing widespread cellular reprogramming after injury could disrupt these networks. There is also the issue of growth control. Regeneration requires significant cell proliferation. In species that regenerate limbs, mechanisms exist to guide and limit this proliferation effectively. In humans, strict control over cell division helps protect against cancer. Tumor suppressor genes monitor cell behavior, stopping growth when abnormalities appear. These safeguards reduce the risk of uncontrolled cell multiplication over a long lifespan. However, they also limit the ability to restart developmental programs in adulthood. If we return to the environments of early Homo species, we see that survival depended not only on physical repair but on cooperation and tool use. A broken limb was serious, yet archaeological evidence suggests that some individuals survived such injuries. Healed fractures found in ancient skeletons indicate that group members likely supported the injured. Social care reduced the evolutionary pressure to develop extreme regenerative abilities. Instead of regrowing a limb, an injured person could adapt, rely on assistance, and continue contributing in other ways. Energy considerations also matter. Regrowing an entire limb would require vast resources. In environments where food was uncertain, allocating such energy might compromise other essential functions. A system that stabilizes injury and relies on social structures for support may have offered a more efficient path. When viewed through this lens, the human inability to regenerate complex structures does not appear as a simple loss. It reflects a shift in priorities. Evolution favored precise organization, long-term stability, cancer resistance, and social cooperation. These traits align with extended lifespan and complex cognition. The cost was reduced flexibility in adult tissues. Thus, the absence of limb regeneration in humans is not an isolated weakness. It is intertwined with the same biological themes that shape slow healing: regulation over speed, stability over dramatic renewal, and collective support over solitary repair. To understand why humans cannot regrow complex structures, it helps to look more closely at what regeneration requires at the cellular level. In animals capable of replacing lost limbs, the injury site does not simply fill with scar tissue. Instead, cells near the wound undergo a remarkable transformation. They loosen their specialized identity and return to a more flexible state. This collection of adaptable cells can then multiply and gradually reorganize into bone, muscle, skin, and nerve, rebuilding the structure in coordinated layers. In adult humans, this kind of reversal is rare. Most of our cells are tightly committed to their roles. A muscle cell is designed to contract. A neuron is designed to transmit signals. These cells operate within a stable framework of gene expression that resists sudden change. This stability protects the organism from chaotic growth. It ensures that tissues maintain their structure and function over decades. Yet it also means that when tissue is lost, the body favors reinforcement over recreation. This restriction is not arbitrary. It reflects the increasing complexity of the human body through evolutionary time. As early Homo species developed larger brains and more refined motor control, the organization of tissues became more intricate. The nervous system in particular depends on precise patterns of connection. Regenerating a limb would require not only rebuilding bone and muscle, but also recreating the exact wiring of nerves to restore sensation and coordination. The margin for error is small. In a species with high reliance on fine motor skills and complex behavior, miswiring could carry serious consequences. Another factor is the long duration of human life. In animals with shorter lifespans, the time window for potential problems caused by cellular reprogramming is limited. In humans, who often live for many decades, even small disruptions in growth regulation can accumulate into disease. Regeneration depends on sustained cell division. Each division carries a chance of mutation. Over a long life, the risk grows. Tight control of cell behavior reduces this risk but restricts flexibility. The fossil record and archaeological findings offer indirect support for this trade-off. Healed injuries show that early humans relied on stabilization rather than regrowth. Bones knit together, joints stiffened, individuals adapted. There is no evidence of lost limbs reappearing. Instead, we see signs of resilience within limitation. The body restored function within structural boundaries, rather than reconstructing lost complexity. Energy also shapes this limitation. Building an entire limb would require immense metabolic resources. In the variable climates of the Pleistocene, where food supply fluctuated and migration was common, dedicating such energy to regeneration may not have been sustainable. A system that repaired enough to restore mobility while preserving overall balance may have been more advantageous. The human strategy, then, appears to favor containment and adaptation over full replacement. Scar tissue forms quickly to close wounds. Bone thickens around fractures. Neural circuits compensate for damage through gradual reorganization rather than wholesale regrowth. These patterns suggest that human biology emphasizes preservation of existing structures over flexible reconstruction. When placed within the broader story of our evolution, this limitation aligns with other themes. Our species invested heavily in cognitive ability, social networks, and extended lifespan. The cost of this investment included stricter growth regulation and reduced regenerative capacity. What we gained in stability and coordination, we traded in flexibility. Thus, the absence of dramatic regeneration in humans is not an isolated weakness. It is woven into a network of adaptations that support long life, complex behavior, and controlled cellular growth. The same safeguards that prevent uncontrolled proliferation also prevent complete structural renewal. In this balance between safety and flexibility, human healing finds its measured pace. If humans did not evolve to regrow entire limbs, it is not because regeneration is impossible in principle. It is because evolution moved in another direction. Over hundreds of thousands of years, our lineage invested in coordination, memory, language, and cooperation. These traits required stable tissues, especially in the nervous system. Stability became more valuable than flexibility. In animals capable of dramatic regeneration, the body can temporarily loosen its structure after injury. Cells reorganize, patterns reform, and lost parts return. This process is guided by signals that resemble those active during early development. In humans, many of those developmental pathways are largely silenced after birth. They are not entirely absent, but they are tightly regulated. Turning them back on in adulthood could disrupt established structures. The human brain illustrates this clearly. Neurons form complex networks during development. These networks depend on precise connections. If adult neurons were free to divide and rewire extensively after injury, the risk of disrupting established circuits would increase. Instead, the brain relies on limited plasticity. Surviving neurons adjust their connections gradually. Function may improve over time, but lost regions are rarely replaced in full. This cautious approach protects identity, memory, and learned behavior across decades. The same principle applies to other tissues. Muscles can grow stronger with use, but they do not regrow entire segments if lost. Skin can close wounds, but it forms scars. Bones can remodel, but they do not recreate perfectly symmetrical shapes after severe damage. These limitations reflect a design oriented toward preserving order. Social evolution also changed the stakes. Early humans did not depend solely on physical perfection to survive. Cooperative groups shared resources and protected vulnerable members. Archaeological evidence shows individuals who survived serious trauma, including limb fractures and degenerative disease. Their survival suggests that adaptation and care compensated for biological limits. Instead of regrowing a limb, a person might use tools differently, rely on assistance, or take on new roles within the group. This social buffer reduced the selective pressure for extreme regeneration. In a solitary species, loss of a limb might mean immediate death. In a cooperative species, survival could continue despite physical limitation. Over time, this changed what traits were favored. Precise coordination and long-term cognitive stability offered greater advantage than the ability to regrow lost tissue. Energy constraints reinforced this shift. Regeneration demands intense metabolic investment. For early humans living with fluctuating food supply, allocating vast resources to rebuild a complex structure may have compromised survival. A system that stabilized injury and relied on gradual adaptation conserved energy for other essential functions. When we ask whether humans lost regeneration, the answer becomes more nuanced. It may be more accurate to say that we narrowed it. We retained regenerative capacity where it was safest and most efficient: skin renewal, liver growth, bone remodeling, while restricting it where risks outweighed benefits. This narrowing aligns with long lifespan, strict growth control, and complex neural organization. Across evolutionary time, these priorities shape the pace and pattern of human healing. Dramatic renewal gave way to controlled repair. Flexibility gave way to regulation. In exchange, humans gained durable tissues, lower cancer risk, and stable cognitive function. The absence of limb regeneration is not an isolated flaw. It is part of a broader strategy that values continuity and order over dramatic reconstruction. In this way, our limited regeneration reflects the same compromise seen throughout the story of human healing. Protect the whole, control growth, endure across time. Among all human tissues, the brain reveals the limits of regeneration most clearly. When neurons are lost due to injury or disease, they are rarely replaced in full. Some small regions retain the ability to produce new neurons, but in most areas of the adult brain, cell division is extremely limited. This restriction may seem like a weakness, yet it reflects a deeper evolutionary choice. The human brain is not only large relative to body size, it is densely interconnected. Billions of neurons form networks that encode memory, language, and learned skills. These networks develop gradually during childhood and adolescence. Connections strengthen or weaken based on experience. By adulthood, patterns of activity are highly organized. If widespread cell division and reorganization occurred after injury, the stability of these networks could be compromised. In species with smaller or less complex nervous systems, regeneration may carry fewer consequences. Simpler circuits are easier to rebuild. In humans, restoring a damaged region would require not only replacing neurons, but also reconnecting them precisely to thousands of other cells. The margin for error is small. A miswired network could disrupt cognition, movement, or emotion. Over evolutionary time, maintaining stable connections may have offered greater advantage than risking large-scale rewiring. This stability aligns with the long lifespan of our species. The brain supports learning accumulated over decades. Knowledge about food sources, migration routes, tool-making techniques, and social relationships contributed to survival. In prehistoric environments, elders may have preserved critical information. A system that preserves neural circuits reliably over time supports this transfer of knowledge. Regenerative flexibility that jeopardizes established networks might reduce overall fitness. There is also a connection between neural stability and cancer risk. Brain cells are highly specialized and generally exit the cell cycle early in development. They do not divide frequently in adulthood. This low rate of division reduces the chance of mutations that could lead to tumors. While brain cancers can occur, their relative rarity compared to other tissues reflects this restrained proliferation. In this case, limited regeneration may protect long-term function. The fossil and archaeological record provides indirect support for the importance of neural stability. Evidence of complex tool use, symbolic behavior, and cooperative hunting suggests that cognitive continuity was crucial. These abilities depended on reliable neural systems. Although skeletal remains cannot reveal brain repair directly, the persistence of cultural patterns across generations implies that cognitive integrity was maintained even when individuals experienced physical injury. When the brain is damaged in modern contexts, recovery often depends on plasticity rather than regeneration. Surviving neurons adjust their connections. Nearby regions may assume new roles. This process can be slow and incomplete, yet it reflects a strategy of adaptation within existing structure rather than replacement of lost tissue. The pace may appear limited, but it preserves continuity. The limits of brain regeneration echo the broader themes of human healing. Stability, regulation, and long-term preservation are prioritized over dramatic renewal. The same evolutionary forces that shaped slow wound repair and controlled cell division shaped the nervous system. In protecting complex networks over decades, the human body accepted constraints on its ability to rebuild from scratch. Thus, when we consider why humans heal slowly compared to other animals, the brain offers a powerful example. Our most valued organ trades regenerative speed for durable structure. The cost is visible in limited repair. The benefit is seen in sustained memory, identity, and learning across a lifetime. Although the adult human brain shows limited regeneration, it is not entirely fixed. Small regions, such as parts of the hippocampus, retain the ability to produce new neurons throughout life. The hippocampus plays a role in memory and spatial navigation. This limited renewal suggests that evolution did not eliminate neural growth completely. Instead, it restricted it to areas where flexibility offered clear benefit without threatening overall stability. This selective pattern reveals an important principle. Regeneration in humans is not absent. It is localized and controlled. In regions where adaptability improves learning and adjustment to new environments, some degree of cell turnover is preserved. In regions responsible for long-term identity, language, and complex motor coordination, division is far more limited. The body differentiates between systems that benefit from renewal and those that require continuity. When the brain is injured, recovery often depends on plasticity rather than replacement. Plasticity refers to the ability of existing neurons to strengthen or weaken their connections. After a stroke or trauma, undamaged areas can sometimes assume partial control of lost functions. This process unfolds gradually. It requires repetition, rehabilitation, and time. The nervous system adapts within its existing architecture rather than constructing entirely new structures. This strategy aligns with the broader human pattern of controlled repair. Rapid neuron proliferation might allow more dramatic recovery, but it would increase the risk of disorganized circuitry. For a species dependent on precise coordination and communication, such risk carries weight. Early humans relied on complex toolmaking, cooperative hunting, and social bonding. These behaviors depended on stable neural networks refined over years. Evolution likely favored brains that preserved established connections even at the expense of regenerative flexibility. The long human lifespan reinforces this pattern. Memory accumulates over decades. Social roles evolve gradually. If neural circuits were frequently dismantled and rebuilt, continuity of knowledge might suffer. By limiting regeneration, the brain protects long-term coherence. The cost is slower or incomplete recovery after injury. The benefit is preservation of accumulated learning. Archaeological findings suggest that individuals with injuries affecting mobility or sensory function sometimes survived long after trauma. Although we cannot directly observe neural repair in fossil records, survival after severe injury implies that adaptation within the nervous system occurred. The body compensated. It reorganized gradually rather than replacing entire regions. This quiet adaptability may have been sufficient in the social context of early human groups. There is also a link between neural restraint and metabolic cost. The brain already consumes significant energy. Sustained cell division would further increase metabolic demand. In environments where food was uncertain, this additional cost might not be sustainable. Restricting regeneration reduces energy expenditure while maintaining essential function. When we consider these layers together, localized neurogenesis, plasticity instead of replacement, long lifespan, energy limits, the pattern becomes clearer. The human brain is designed for continuity more than reconstruction. It adapts slowly within its framework rather than restarting from the beginning. In this way, neural healing mirrors the broader human approach to repair. It is cautious, regulated, and dependent on time. It values preservation of identity over dramatic renewal. What appears as limited regeneration is part of a larger evolutionary balance between flexibility and stability, shaped by the demands of a long-lived, socially complex species. When we follow the story of the brain to its quiet conclusion, we begin to see that limited regeneration is not an accident. It is part of a larger pattern in human evolution. The brain did not evolve primarily to rebuild itself. It evolved to preserve patterns of memory, language, social bonds, and learned skill. In a species whose survival depended on shared knowledge, continuity mattered more than renewal. Early humans lived in small groups where information was critical. Knowing which plants were edible, which animals were dangerous, where water could be found during dry seasons, or how to shape a tool from stone were not simple instincts. They were learned behaviors, passed across generations. A brain that maintained stable circuits over decades could store and transmit such knowledge reliably. Frequent structural turnover might have threatened that stability. When injury occurred, the nervous system responded within these limits. Rather than replacing lost neurons wholesale, it adjusted surviving networks. Connections strengthened or weakened. Adjacent regions sometimes took on new roles. This process required time and repetition. It depended on social interaction, practice, and gradual adaptation. In prehistoric contexts, this slow reorganization may have been sufficient. The group provided support while the injured individual adjusted. The fossil record does not reveal neural tissue directly, but skeletal evidence suggests that individuals with significant physical injuries often survived long after trauma. This implies that their cognitive function remained intact enough to participate in group life. The stability of the brain likely contributed to this resilience. Even when the body was altered, the continuity of memory and identity endured. There is also a deeper biological safeguard involved. Neurons are among the most specialized cells in the body. Once formed, they rarely re-enter the cell cycle. This reduces the chance of mutations that could lead to tumors. Brain cancers, while serious, are relatively rare compared to malignancies in tissues with high turnover. Limited division protects neural integrity over a long lifespan. In a species that can live for many decades, this restraint becomes valuable. Energy once again plays a role. The brain's high metabolic demand leaves little room for extensive regeneration. Building new neural networks from scratch would require substantial resources. In ancient environments with fluctuating food supply, such investment may not have been practical. Instead, gradual adjustment within existing structures offered a more economical path. This cautious design reflects the broader human strategy. Across tissues, we see preference for regulation, reinforcement, and adaptation over dramatic renewal. The brain exemplifies this principle most clearly. It preserves organization at the cost of flexibility. It protects accumulated experience at the cost of rapid repair. When we ask why humans heal slowly compared to other animals, the nervous system provides a powerful answer. Our most complex organ favors stability across decades. It guards against uncontrolled growth. It maintains identity and memory. The price is limited regeneration. Yet this price is intertwined with the very traits that define our species: long life, culture, cooperation, and learning. In the quiet architecture of the brain, we can see how evolution shaped human healing not for spectacle, but for endurance. The path chosen was careful, controlled, and steady. It allowed knowledge to persist, even when bodies bore marks of injury. When we think about healing, we often imagine it as something that happens only inside the body. Cells divide, tissue closes, pain slowly fades. Yet for humans, healing has never been purely biological. From the earliest chapters of our history, recovery was shaped not only by cells and proteins, but by other people. The human body heals within a social environment, and this environment has influenced how our biology evolved. In many animal species, severe injury can mean isolation and death. A broken limb in a solitary predator may prevent hunting. A wound may lead to vulnerability without assistance. In such contexts, strong pressure exists for rapid individual recovery. Survival depends almost entirely on the injured body's own speed and capacity. Humans, however, evolved within cooperative groups. Early Homo species shared food, defended territory together, and cared for offspring collectively. This social structure altered the consequences of injury. Archaeological discoveries offer quiet evidence of this pattern. Fossilized skeletons have been found showing healed fractures, severe dental infections, and even conditions that would have limited mobility for extended periods. In some cases, individuals survive for years after such injuries. A broken femur, for example, requires months to heal and makes walking nearly impossible without assistance. The survival of individuals with healed femoral fractures suggests that others supported them during recovery. This social support reduces selective pressure for extreme biological regeneration. If the group can compensate for temporary weakness, the body does not need to repair itself at maximum speed to ensure survival. Instead, healing can proceed steadily while the injured individual remains protected. Cooperation becomes a biological buffer. The long childhood of humans reinforces this dynamic. Children depend on adults for many years. Adults in turn rely on social bonds for protection and shared labor. Injury does not remove an individual's value to the group. Even with limited mobility, a person may contribute knowledge, tool-making skill, childcare, or planning. This continuity of role reduces the evolutionary cost of slower physical repair. There is also a psychological dimension. Social connection influences stress levels, and stress hormones affect immune function. Chronic stress can slow wound healing by altering inflammatory responses and reducing tissue regeneration. In supportive social environments, stress may be moderated, indirectly aiding recovery. Although prehistoric stressors differed from modern ones, the presence of stable social bonds likely shaped physiological responses over time. From an evolutionary perspective, the interplay between social structure and biology created a feedback loop. Groups that supported injured members increased overall survival. Individuals who could rely on assistance did not require dramatic regenerative abilities. Over generations, traits favoring cooperation and stable healing patterns were reinforced. This does not mean that early human life was gentle. Injuries could still be fatal. Infection remained a constant threat. Yet the existence of healed skeletal remains across different archaeological sites indicates that recovery often succeeded within a communal context. The body's measured pace of repair fit within a network of social resilience. When we ask why humans heal slowly compared to other animals, the answer extends beyond cells. It reaches into the structure of early human communities. Healing unfolded not only in tissue but in shared labor and protection. Our biology evolved alongside cooperation. The group absorbed some of the risk that, in other species, would demand rapid individual regeneration. Thus, human healing is partly a collective phenomenon. The body repairs within limits, while the community bridges gaps. This partnership between biology and society shaped the tempo of recovery long before written history. If healing in humans unfolds within a social context, then the meaning of survival changes. A wounded animal alone must depend entirely on its own speed of recovery. A wounded human in a cooperative group does not face the same isolation. Food can be shared, shelter can be provided. Protection can continue even if one member is temporarily weakened. This difference alters the evolutionary pressure placed on the body. Archaeological sites across Africa and Eurasia have revealed skeletons bearing clear signs of healed trauma. Some individuals show evidence of broken ribs, damaged limbs, or degenerative joint disease that would have limited movement for long periods. In certain cases, injuries appear severe enough that independent survival would have been unlikely. Yet these individuals lived long enough for bones to knit and remodel. The time required for such healing suggests sustained support from others. This pattern indicates that early human groups did more than tolerate injured members, they invested in them. That investment changes what biology must accomplish alone. If the group can protect and feed an injured individual, the body does not need to regenerate at extraordinary speed. Steady repair becomes sufficient. The cooperative nature of human life extends beyond emergency care. Shared hunting, food distribution, and division of labor create redundancy. If one individual cannot perform a task temporarily, others can compensate. In solitary species, such redundancy is rare. In cooperative species, it becomes a buffer against individual limitation. This social buffering likely influenced the direction of natural selection. Traits that favored group cohesion, communication, and empathy would increase overall survival. Over generations, the advantage of strong cooperation may have outweighed the need for rapid biological regeneration. A species that heals together does not require each body to operate as an isolated survival machine. There is also a demographic dimension. Humans evolved extended family structures. Grandparents, parents, siblings, and unrelated group members contributed to collective resilience. The survival of an injured adult might still benefit offspring through knowledge, transfer, or care. Even if physical capacity was reduced, cognitive and social contributions remained valuable. This reduces the evolutionary cost of slower physical recovery. Modern research supports the idea that social support influences healing. Studies show that individuals with strong social connections often experience better recovery outcomes after illness or surgery. Stress levels, immune function, and hormonal balance all interact with social environment. While prehistoric conditions were different, the fundamental link between connection and physiology likely existed. The tempo of human healing, therefore, reflects more than cellular biology. It reflects the rhythm of group life. In early Homo communities, injury did not automatically mean abandonment. Care and cooperation provided time for gradual repair. This time reduced the necessity for rapid regeneration. When we consider why humans heal slowly compared to certain animals, we must include this layer of explanation. Our ancestors did not rely solely on tissue repair to survive. They relied on one another. The body could afford to proceed carefully because the group provided protection during vulnerability. Over many generations, this partnership between social structure and biology shaped the pace of human healing. Slow recovery, in this context, is not purely a limitation. It is part of a broader adaptive system that includes cooperation, shared labor, and emotional bonds. The body heals within community, and community changes what healing must achieve alone. If we step back and look at human healing through the lens of community, the picture becomes quieter but clearer. The human body does not repair itself in isolation from the social world. From the earliest bands of hunter-gatherers to later settled communities, survival has depended on shared effort. This shared effort shaped not only culture but also biology. In small prehistoric groups, daily life required cooperation. Hunting large animals demanded coordination. Gathering plant foods required knowledge and memory passed between generations. Fire needed tending. Children required constant care. Within this web of interdependence, an injured individual did not necessarily become useless. Roles could shift. A person with limited mobility might contribute through planning, teaching, tool preparation, or childcare. The value of an individual extended beyond immediate physical strength. This flexibility in social roles altered evolutionary pressure. In solitary animals, severe injury can remove an individual from reproduction quickly. Natural selection may therefore favor rapid regeneration where possible. In humans, reproduction and survival were supported by group networks. Even if one member was temporarily impaired, the group could absorb the cost. Over generations, this buffering effect reduced the advantage of extreme biological repair. Archaeological findings reinforce this interpretation. Sites dating back tens of thousands of years show individuals who survived injuries that would have required extended recovery. Some skeletons display healed fractures of weight-bearing bones, suggesting months of limited mobility. Others show signs of degenerative disease. The survival of these individuals implies sustained care. It also suggests that the group invested resources in those who could no longer perform all tasks independently. This investment carries biological consequences. If community support increases survival during vulnerability, the body does not need to evolve extraordinary regenerative capacity to achieve reproductive success. Instead, it can prioritize traits that enhance cooperation, communication, and long-term stability. A slower, regulated healing process fits within this framework. There is also a psychological dimension that may have influenced physiology over time. Human bonding reduces stress responses. Lower chronic stress can support immune function and tissue repair. In ancestral environments, strong social bonds may have moderated the hormonal signals that influence healing. While the details of prehistoric stress cannot be measured directly, the consistent importance of social cohesion in human evolution suggests an intimate connection between community and recovery. Across generations, the interplay between biology and society created a unique rhythm of healing. The body repaired within limits shaped by energy, cell regulation, and longevity. The group provided protection and continuity. Together, these elements formed a system where survival did not depend solely on rapid regeneration. When we ask why humans heal slowly compared to other animals, this social layer completes part of the answer. Our ancestors did not face injury alone. They healed within a network of relationships. This network softened the evolutionary demand for speed. It allowed the body to remain cautious, controlled, and stable across decades. In this sense, human healing is partly collective. The scars on one body were often carried by many hands that helped it endure. Over time, cooperation became woven into the biology of recovery. The pace of healing reflects not only cellular processes but also the quiet strength of shared life. In the prehistoric world, the greatest threat following injury was often not the wound itself but infection. Before the discovery of microbes, before antiseptics or antibiotics, the environment was filled with invisible organisms. Soil contained bacteria, animal bites introduced pathogens, water sources carried microorganisms. A small cut could become dangerous if bacteria entered the bloodstream. In this setting, the speed and strength of the immune response mattered more than cosmetic restoration. Early humans lived in close contact with the natural world. They butchered animals, handled raw hides, and walked barefoot across rough ground. Stone tools, though sharp and effective, could easily break the skin. The absence of sterile conditions meant that every wound was exposed to microbial life. Natural selection, therefore, favored immune systems that reacted quickly and decisively. Even if this reaction caused swelling and delayed visible healing, it increased the chance of survival. The inflammatory response described earlier is central to this defense. Once tissue is damaged, immune cells flood the area to neutralize invading microbes. Blood vessels dilate, bringing more cells and nutrients. Chemical signals recruit additional defenders. This intense activity may slow the structural rebuilding of tissue, yet it creates a hostile environment for pathogens. In prehistoric contexts, containing infection was often the difference between recovery and death. Archaeological evidence provides glimpses of this ancient struggle. Skeletal remains sometimes show signs of osteomyelitis, a bone infection that can leave characteristic marks. Other remains reveal healed lesions or abscesses. These traces indicate that infections occurred but were sometimes overcome. The immune system did not prevent every infection, yet it succeeded often enough to allow individuals to survive and reproduce. The human approach to healing, therefore, reflects this constant negotiation with microbes. The body does not prioritize rapid tissue regeneration if infection remains unresolved. Instead, it ensures that bacterial threats are controlled before full repair proceeds. This sequence can extend the visible duration of healing, but it reflects a logical priority: eliminate danger first. Fire and plant knowledge also influence this balance. Early humans used fire not only for warmth and cooking, but likely for cleaning wounds and tools. Certain plants possess antimicrobial properties. While the details of prehistoric medical practices are difficult to reconstruct, ethnographic studies of more recent hunter-gatherer societies show that plant-based remedies were common. These cultural adaptations complemented biological defenses, but they did not replace them. Energy constraints again played a role. Fighting infection is metabolically demanding. Fever increases energy use. Immune cells consume fuel at high rates. In environments with fluctuating food supply, the body could not afford to sustain prolonged infection battles lightly. Thus, the immune system evolved to act strongly but with regulatory mechanisms to prevent endless activation. When we consider why humans heal slowly compared to other animals, infection risk provides a critical piece of the puzzle. In a world without modern medicine, survival required prioritizing defense over speed. The body's cautious, stepwise repair process ensured that pathogens were contained before tissue rebuilding advanced fully. This ancient pattern remains embedded in our physiology. Even today, wounds must be clean before they heal efficiently. Antibiotics may accelerate recovery by reducing bacterial load, but the underlying sequence remains the same. The immune system clears threats. Only then does regeneration continue. Slow healing in this sense reflects the long history of human life in a microbe-rich world where infection was a constant shadow. If infection was the central danger after injury, then the tempo of healing must be understood as a response to that danger. The body cannot rebuild safely while microbes remain active. A tissue that regenerates too quickly over contaminated material can trap bacteria inside, allowing infection to spread beneath the surface. For this reason, human healing follows a deliberate sequence: cleanse, defend, then reconstruct. This order may slow visible recovery, yet it protects long-term survival. In prehistoric environments, wounds were rarely clean. Stone blades, though sharp, were not sterile. Animal bites introduced saliva, filled with bacteria. Even simple cuts from plants exposed tissue to soil organisms. Under these conditions, immune cells had to work intensely before rebuilding could proceed. Neutrophils and macrophages engulfed invading microbes and cleared debris. Only after this stage diminished did fibroblasts lay down collagen and begin structural repair. This pattern is reflected in modern medicine. Surgeons clean wounds thoroughly before closing them. In contaminated injuries, doctors may leave a wound partially open to drain and prevent trapped infection. These practices mirror ancient biological logic. The body evolved to treat infection control as the first priority. Rapid closure without adequate cleansing would increase risk. Archaeological findings support the prevalence of infection in early human populations. Skeletal evidence of bone lesions, dental abscesses, and joint infections suggests repeated exposure to pathogens. Yet many individuals survive these episodes. Their bones show signs of healing around infection sites. This indicates that the immune system often succeeded in containing bacterial threats long enough for recovery to occur. Environmental conditions intensified this challenge. Warm climates accelerate bacterial growth. Water sources may carry parasites and microbes. Animal carcasses, handled during hunting and butchering, introduced additional exposure. In such contexts, a cautious and thorough immune response offered advantage. Even if healing took longer, survival odds improved when infection was fully addressed. The energy cost of fighting infection also shaped the pace of recovery. Fever raises metabolic demand. Immune cells consume glucose rapidly. In times of food scarcity, Sustaining prolonged immune activity could weaken the body. Therefore, regulatory systems evolved to balance intensity and duration. The inflammatory response rises quickly but must taper once pathogens are neutralized. Only then can tissue repair advance without excessive collateral damage. Cultural adaptations gradually supplemented biological defenses. The controlled use of fire may have reduced microbial load on tools and wounds. Knowledge of certain plants with antiseptic properties likely developed over generations. These practices did not eliminate infection risk, but may have improved outcomes. Still, the core sequence remained embedded in physiology. Contain threat before rebuilding. When we compare humans to animals with faster regeneration, we must remember that many of those animals inhabit environments or follow life strategies where infection dynamics differ. Some species live in aquatic settings with different microbial exposure. Others have immune systems calibrated differently due to shorter lifespans. Humans, with extended life and complex social environments, evolved a healing process heavily shaped by microbial risk. Thus, the slower pace of human healing reflects not weakness but prioritization. The body ensures that pathogens are neutralized before investing energy in reconstruction. This layered response, forged in environments rich with microbes, continues to guide recovery today. Beneath the surface of every wound lies a history of negotiation between tissue and infection, a history that shaped the measured rhythm of human repair. If infection shaped the order of healing, it also shaped the body's tolerance for imperfection. In a world where microbes were constant and invisible, the primary objective after injury was containment. The body did not need to restore skin or bone to their original form immediately. It needed to prevent bacteria from spreading deeper. Once that barrier was secure, gradual rebuilding could continue with less urgency. This sequence left traces not only in tissue but in the archaeological record. Some ancient skeletons show evidence of chronic infection that eventually stabilized. Bone surfaces display irregular thickening where the immune system walled off bacteria. These irregularities suggest that the body sometimes chose isolation over elimination. Rather than eradicate every pathogen instantly, it encased the threat within fibrous or bony tissue. This strategy preserved life even if it altered structure. Such containment aligns with the broader human pattern of controlled repair. The immune system, while powerful, does not aim for total destruction of every microorganism. Many microbes coexist harmlessly within the human body. The goal is balance. After injury, inflammation rises to suppress harmful invaders, then recedes to avoid excessive damage. The visible delay in healing often reflects this cautious calibration. Environmental variability reinforced this need for moderation. In dry seasons, wounds might heal more quickly due to lower microbial activity. In wetter conditions, infection risk would rise. The body's flexible response, strong defense, followed by gradual reconstruction, allowed adaptation across changing climates. Over generations, this approach proved reliable enough to sustain survival. Energy constraints once again intersect with infection control. Fighting microbes demands fuel. Prolonged fever and immune activation draw on stored resources. In prehistoric contexts, where caloric intake fluctuated, the body had to conserve energy carefully. Excessively aggressive regeneration combined with intense immune activity could deplete reserves. A phased approach spread energy use over time. The rise of cultural practices gradually influenced this balance. Fire, which early humans mastered hundreds of thousands of years ago, likely reduced bacterial exposure in food and possibly in wound care. Some plant substances with antimicrobial properties may have been applied to injuries. These innovations supplemented biology but did not replace it. The underlying physiological sequence, defense first, repair second, remained unchanged. Even today, this ancient logic persists. Modern medicine emphasizes wound cleaning, infection prevention, and careful monitoring before complete closure. Antibiotics accelerate microbial control, yet the body still follows its inherited order. Infected wounds heal slowly because inflammation must remain active until pathogens are neutralized. Thus, the tempo of human healing reflects a long negotiation with the microbial world. Our ancestors lived in environments where infection was a daily risk. Survival favored bodies that prioritize containment and regulation over speed. The scars and extended recovery times that follow injury today echo that ancient priority. When we ask why humans heal slowly compared to other animals, infection stands as a central explanation. The body moves deliberately, ensuring safety before restoration. This caution, shaped by countless encounters with unseen microbes, became embedded in our biology. It is not a flaw, but a reflection of the environments in which our species endured. If we move from wounds and infection to the level of individual cells, another limit to human healing becomes visible. Every time tissue repairs itself, cells must divide. Skin cells multiply to close a cut. Bone cells reproduce to bridge a fracture. Blood vessels expand through new cellular growth. Division is essential to repair, yet division also carries risk. With each cycle of copying DNA, there is a possibility of error. The human body has evolved multiple layers of protection to monitor this process. Genes act as supervisors, ensuring that cells divide only when necessary and stop when their task is complete. Among these safeguards are tumor suppressor mechanisms that trigger cell death if abnormal growth is detected. These systems reduce the likelihood of cancer. However, they also restrict how aggressively tissues can regenerate. In species with shorter lifespans, the balance between regeneration and cancer risk may tilt differently. If an animal typically lives only a few years, the long-term consequences of minor DNA errors may never appear. Rapid growth and quick regeneration can proceed without severely compromising reproductive success. Humans, by contrast, often live for many decades. The cumulative effect of small cellular mistakes increases over time. Evolution therefore favored stricter control over cell division in long-lived species. This strict regulation shapes the pace of healing. Cells involved in repair must respond to chemical signals indicating damage. Once the wound is stabilized, these signals diminish. Cells return to a resting state. The body avoids prolonged or excessive growth, even if more rapid proliferation could restore tissue more quickly. The price of speed might be instability. Archaeological evidence indirectly reflects this long-term caution. Ancient skeletons often show healed injuries rather than signs of malignant bone growth. While cancer certainly existed in prehistoric populations, its relative rarity in skeletal remains may partly reflect both shorter average lifespans and careful biological regulation. The same regulatory systems that slow regeneration help prevent uncontrolled growth. Energy also intersects with this risk management. Rapid cell division requires significant metabolic input. DNA synthesis, protein production, and membrane construction all consume fuel. In environments where food supply fluctuated, allocating excessive energy to tissue growth could weaken other vital functions. A measured pace conserved resources while maintaining safety. Modern medical science highlights this trade-off clearly. Treatments that stimulate cell division, such as certain growth factors, can enhance repair but must be used cautiously due to cancer risk. Conversely, therapies that suppress cell proliferation may reduce tumor formation but slow wound healing. These opposing effects demonstrate that regeneration and cancer prevention exist on a continuum rather than as separate systems. The human body sits at a point on this continuum that prioritizes long-term control. It allows repair, but within boundaries. It monitors growth carefully. When damage occurs, it responds deliberately, balancing reconstruction against the risk of runaway division. This equilibrium evolved over thousands of generations in organisms that expected to live long enough for cellular errors to matter. When we ask why humans heal slowly compared to other animals, cancer risk forms a quiet but powerful part of the answer. The body does not simply rebuild at maximum speed. It weighs every act of division against decades of future life. The pace may appear cautious, yet that caution protects the integrity of tissues across time. In choosing regulation over rapid renewal, the human organism preserved longevity at the expense of dramatic regeneration. To understand how cancer risk shapes healing, we must look more closely at how cells decide to divide. Within each cell lies DNA, the long sequence of instructions that guides growth and function. When a cell prepares to divide, it must copy this sequence with extraordinary precision. Specialized enzymes proofread and repair errors, yet no system is flawless. Over time, small changes can accumulate. Most remain harmless. A few may alter the signals that tell a cell when to stop dividing. In humans, Layers of protective mechanisms evolve to reduce this risk. Proteins monitor DNA integrity. If damage is detected, the cell may pause to repair it. If the damage is severe, the cell may self-destruct through a controlled process known as apoptosis. These safeguards reduce the likelihood of tumors forming from uncontrolled proliferation. However, they also slow processes that depend on rapid cell multiplication, including tissue regeneration. In shorter-lived species, the evolutionary balance may differ. If the typical lifespan spans only a few seasons, the cumulative chance of dangerous mutations emerging remains relatively small. In that context, rapid repair may offer more immediate benefit than strict long-term regulation. For humans whose ancestors increasingly lived into extended adulthood, even small increases in mutation risk could affect survival and reproduction over decades. The fossil record offers subtle support for this long-term perspective. While skeletal remains cannot capture all cancers, the relative scarcity of clear malignant bone lesions compared to the frequency of healed fractures suggests that severe bone tumors were not widespread. This may partly reflect demographic patterns, but it also aligns with the presence of robust cellular regulation systems. Controlled growth protected long-term stability. Modern research reinforces this connection. Genes that act as tumor suppressors, such as those involved in halting abnormal division, are essential for preventing cancer. Yet when these regulatory pathways are highly active, they can limit regenerative capacity. In laboratory studies, altering these pathways can enhance tissue growth, but often increases cancer risk. The biological tension is evident. What accelerates repair may also destabilize control. This tension extends beyond skin and bone. Internal organs follow similar rules. The liver can regenerate more effectively than many other tissues, yet even this capacity has limits. Prolonged stimulation of liver growth can increase cancer risk. The body therefore balances regenerative signals with inhibitory feedback, maintaining a steady pace rather than unchecked expansion. From an evolutionary standpoint, this balance makes sense for a species built for endurance. Humans did not evolve to live only long enough to reproduce once. Cooperative social structures and extended care of offspring favored survival across decades. A system that minimized catastrophic cellular errors would enhance overall fitness. Slower regeneration became part of this protective framework. Energy once again intersects with regulation. DNA repair, cell cycle checkpoints, and programmed cell death all require metabolic investment. These processes consume resources but safeguard integrity. In unpredictable ancestral environments, conserving long-term function likely outweighed the benefits of rapid but risky repair. Thus, human healing unfolds under strict supervision. Cells divide, but only within boundaries. Growth signals activate, then recede. Repair advances, then stabilizes. The pace may seem modest compared to certain animals, yet it reflects a deliberate compromise between regeneration and safety. When we ask why humans heal slowly compared to other animals, cancer prevention emerges as a central theme. The body's caution is not hesitation but protection. It limits proliferation to preserve life across decades, accepting slower repair in exchange for stability and reduced risk. If we follow the thread of cell division to its deeper conclusion, a quiet pattern appears. The same mechanisms that slow human healing are the ones that guard against internal chaos. Every time tissue repairs itself, a decision is made at the microscopic level. Signals activate genes, proteins assemble, DNA is copied. Then, just as importantly, other signals intervene to limit the process. Repair does not run unchecked. It stops when enough stability has been restored. In a long-lived organism, this restraint is essential. A body that expects to function for many decades cannot allow frequent bursts of uncontrolled growth. Even small errors in DNA replication, if multiplied over time, can lead to malignancy. Tumors arise when growth signals persist beyond their proper limits. Therefore, evolution favored systems that interrupt proliferation once structural integrity returns, even if that means tissue is not perfectly reconstructed. This principle can be observed in the way scars form and stabilize. After collagen is deposited and the wound closes, remodeling continues slowly, but active cell division declines. The body does not continue rebuilding indefinitely in pursuit of perfection. It accepts a structurally sound outcome and redirects energy elsewhere. The scar becomes a boundary, a reminder that repair has reached a safe stopping point. In species with shorter lifespans, the calculus differs. The cumulative burden of small genetic errors is less likely to manifest within a limited lifespan. Faster regeneration may pose less long-term threat. In humans, where extended survival became increasingly common, even modest increases in mutation risk could affect health decades later. The balance shifted toward caution. Archaeological remains show that many ancient individuals lived long enough for age-related changes to appear in their skeletons. Joint wear, healed fractures, and degenerative conditions indicate prolonged survival. These records imply that long-term tissue integrity mattered. A body prone to frequent uncontrolled growth would compromise that endurance. The quiet success of regulated healing contributed to survival across time. There is also an interaction between cancer prevention and immune surveillance. The immune system not only fights infection but also detects abnormal cells. Persistent, excessive regeneration could overwhelm these surveillance mechanisms. By limiting proliferation, the body reduces the burden on immune monitoring. Slow repair becomes part of a broader defensive architecture. Energy considerations again reinforce this design. DNA repair pathways, checkpoint proteins, and programmed cell death mechanisms consume resources. These safeguards are metabolically costly, yet they protect against catastrophic outcomes. In environments where food availability fluctuated, investing energy in regulation rather than rapid reconstruction may have offered greater overall benefit. When we place these elements together, longevity, DNA surveillance, immune monitoring, metabolic limits, the measured pace of human healing appears less mysterious. It is embedded within a system designed to preserve order over decades. The body does not chase flawless regeneration if doing so would increase risk. It repairs sufficiently, then restrains further growth. Thus, the slow tempo of human healing reflects a protective compromise. It accepts visible imperfection to avoid invisible instability. It limits regeneration to safeguard long-term survival. In doing so, it aligns with the broader evolutionary pattern that shaped our species. Stability over spectacle, endurance over rapid renewal. When we compare ourselves to animals that regenerate dramatically, we are witnessing different life strategies rather than simple superiority or deficiency. Human healing carries the imprint of long life and careful regulation. The same systems that slow repair quietly protect us from dangers that accumulate across time. If we shift from deep prehistory to the present day, a quiet contrast appears. The human body that heals slowly today is the same body shaped in ancient landscapes. Yet the environment around it has changed dramatically. Modern medicine can clean wounds, control infection, replace lost blood, and even transplant organs. Antibiotics reduce bacterial threats that once proved fatal. Surgical techniques repair fractures with precision unimaginable to early humans. Still, the pace of natural healing inside the body remains much the same. This contrast highlights an important idea. Our biological systems evolved under conditions of scarcity, microbial exposure, and social cooperation. They did not evolve in sterile hospitals with steady food supply and climate control. When we observe that humans heal slowly compared to certain animals, we are observing an ancient program operating in a modern world. Modern medical practice often supports or accelerates stages of healing that were once limiting. Cleaning and disinfecting a wound reduces the need for prolonged inflammation. Antibiotics assist the immune system in clearing bacteria. Nutritional support ensures adequate protein and micronutrients for tissue repair. Surgical stabilization of fractures allows bone remodeling to proceed more efficiently. These interventions do not fundamentally change how cells divide or how scars form, but they remove obstacles that once prolonged recovery. Even so, the core sequence remains. Bleeding must stop. Inflammation must control infection. Collagen must be deposited. Tissue must remodel gradually. Doctors cannot force skin to regenerate without scar in the way certain animals can. They work within the constraints of human biology. This persistence reveals how deeply embedded those constraints are. In the past, survival after serious injury often depended on group support. Today, medical systems extend that support at a societal level. Hospitals, rehabilitation centers, and long-term care facilities function as modern extensions of ancient cooperation. They buffer vulnerability while the body repairs at its inherited pace. Technology supplements, but does not replace biology. There is also a new dimension in the modern world: lifespan. Many humans now live well beyond the ages common in prehistoric times. This extended lifespan exposes the long-term effects of cellular regulation and repair. Chronic diseases, age-related degeneration, and cumulative tissue wear become visible. The same cautious systems that once protected against cancer and infection operate across an even longer timeline. Modern research attempts to enhance healing through growth factors, stem cell therapies, and regenerative medicine. These approaches aim to stimulate tissue repair more dramatically, yet each carries questions about long-term safety. Increasing cell proliferation can improve regeneration, but it may also increase cancer risk. The ancient balance between repair and control still shapes scientific caution. Seen in this light, the slow pace of human healing is not outdated or malfunctioning. It is consistent with a body designed under evolutionary pressures very different from modern ones. Medicine can assist, optimize, and protect, but it works alongside inherited limits. When we ask why humans heal slowly compared to other animals, the answer does not disappear in the age of technology. Instead, it becomes clearer. Our bodies still operate according to ancient trade-offs: infection control before speed, regulation before proliferation, stability before spectacle. Modern life may soften the consequences of these trade-offs, but it does not erase them. The body that closes a wound today is following instructions written long before hospitals existed. The tools have changed. The cellular logic has not. Modern medicine has altered the risks surrounding injury, but it has not rewritten the underlying biology. When a surgeon closes a wound with precision or stabilizes a fracture with metal plates, the body still must perform the same cellular work it has always performed. Blood must clot, immune cells must clear debris, fibroblasts must lay down collagen, bone cells must bridge the gap and slowly remodel. Technology can guide the process, but it cannot bypass the fundamental rhythm shaped by evolution. One of the clearest examples of this interaction appears in infection control. Antibiotics reduce bacterial load, allowing inflammation to subside more quickly. In ancient environments, the immune system alone carried this burden. Today, pharmaceutical assistance shortens the defensive phase. Yet once infection risk is reduced, tissue rebuilding still follows its inherited pace. Skin does not regenerate without scar simply because antibiotics are present. The biological limits remain. Nutrition provides another example. Modern societies with consistent food supply allow for steady intake of protein, vitamins, and minerals essential for healing. Vitamin C supports collagen synthesis. Zinc contributes to immune function. Adequate protein supplies amino acids for tissue construction. In prehistoric times, these nutrients fluctuated with seasons and hunting success. Today, stable nutrition can optimize healing within natural constraints. It does not transform human tissue into amphibian-like regenerative systems, but it ensures that the available capacity functions efficiently. Rehabilitation practices reveal the importance of gradual remodeling. After injury, controlled movement strengthens tissue alignment. Physical therapy helps bone and muscle adapt to stress. This reflects an ancient principle: tissues remodel in response to use. Modern rehabilitation accelerates recovery by guiding this remodeling carefully. Yet even here, progress is incremental. The body integrates stress signals over weeks and months. Advances in regenerative medicine, including stem cell research, attempt to expand the limits of repair. Scientists explore ways to stimulate tissue growth or reprogram cells to regain flexibility. These efforts are promising but cautious. Every attempt to enhance proliferation must consider cancer risk and long-term stability. The ancient balance between growth and control still governs decision-making. Extended lifespan in the modern era further exposes the importance of regulation. People now live long enough to experience cumulative effects of cellular aging. Wound healing may slow with age due to changes in immune response and reduced cellular division capacity. These changes reflect the same regulatory systems that protected earlier generations from uncontrolled growth. Longevity magnifies the trade-offs embedded in our biology. The presence of advanced healthcare systems can create the impression that human healing has fundamentally improved. In reality, medicine reduces external threats and supports internal processes. It does not replace the evolutionary logic underlying repair. The body continues to weigh energy expenditure, infection risk, and mutation control as it rebuilds tissue. When we examine this modern context, the question of slow healing gains clarity rather than contradiction. Humans heal at a pace aligned with long-term safety. Medicine shortens some phases and protects against complications, but it operates within inherited boundaries. The ancient program of cautious repair continues, even in sterile operating rooms. Thus, modern life does not erase the reasons behind slow human healing, it highlights them. The more we intervene, the more visible the underlying constraints become. Our cells still follow the same guarded steps that once preserved survival in environments without antibiotics or surgical tools. When we look closely at the relationship between modern medicine and ancient biology, something subtle becomes clear. Medicine does not make the human body regenerate like a salamander. It does not erase scars at the cellular level. What it does is remove obstacles that once made slow healing more dangerous. It lowers the cost of our biological compromises without rewriting them. In prehistoric times, a deep wound carried several risks at once: blood loss, infection, limited mobility, and reduced access to food. The body addressed these risks sequentially and cautiously. In modern hospitals, bleeding can be controlled rapidly with surgical tools. Infection can be treated with antibiotics. Fractures can be aligned and stabilized precisely. These interventions reduce uncertainty. They give the body a safer environment in which to proceed at its natural pace. Yet even with these supports, healing still unfolds in stages that cannot be rushed beyond certain limits. A bone still requires weeks to knit together because osteoblast cells must produce matrix, deposit minerals, and remodel gradually in response to stress. Skin still forms scar tissue through collagen deposition because the regulatory systems governing fibroblast activity remain unchanged. Neural tissue still adapts through plasticity rather than wholesale replacement. The persistence of these patterns reveals that the slow tempo of human healing is not merely a relic of primitive conditions. It is part of a deeply integrated system balancing longevity, cancer prevention, immune control, and energy management. Medicine may reduce infection and improve nutrition, but it does not remove the long-term risks associated with excessive cell proliferation. For this reason, attempts to dramatically accelerate regeneration remain cautious and experimental. Extended lifespan in modern societies makes these trade-offs even more visible. People now live long enough to experience the cumulative effects of aging tissues. Wound healing may become slower with age due to reduced stem cell activity and changes in immune response. These shifts reflect the same regulatory frameworks that once protected younger adults from uncontrolled growth. Longevity magnifies the importance of restraint. In this context, modern healthcare can be seen as a continuation of ancient cooperation. Where small bands once supported injured members, now entire medical systems provide collective assistance. Hospitals, clinics, and rehabilitation programs extend the social buffer that has long characterized human survival. They allow the body to heal within its inherited limits, while minimizing external threats. The contrast between technological progress and biological continuity invites reflection. Despite advances in science, the human body still follows instructions shaped in prehistoric landscapes. It closes wounds carefully, it regulates growth strictly, it rebuilds slowly. The tempo of healing has not fundamentally changed because the underlying logic remains protective. When we ask why humans heal slowly compared to other animals, modern medicine does not overturn the answer. Instead, it clarifies it. Our healing speed reflects a balance designed for long-term stability rather than rapid replacement. Technology may ease the journey, but it does not alter the evolutionary compromise at the core. Thus, even in an age of advanced treatment, the ancient rhythm persists. The body repairs with caution, mindful of risks that unfold over decades. In the quiet space between injury and recovery, we can still hear echoes of the environments in which our species first learned to survive. When we gather together all the threads, energy limits, infection risk, cancer prevention, neural stability, and social cooperation, a single pattern emerges. Human healing is not designed for spectacle. It is designed for survival across time. Each layer of restraint that slows visible recovery also protects something larger. The body does not rush to rebuild because it must preserve balance within a system expected to endure for decades. From the earliest Homo populations moving through African landscapes to present-day societies, the same biological principles have persisted. Cells divide under supervision. Inflammation rises and falls under regulation. Scar tissue forms quickly to secure safety. Remodeling continues quietly beneath the surface. None of these steps are accidental. They reflect accumulated decisions shaped by millions of years of selection. In evolutionary terms, healing is always part of a trade-off. To regenerate perfectly would require flexibility in adult cells, extensive proliferation, and high metabolic investment. These features increase risk of mutation, risk of instability, risk of exhausting energy reserves. Humans, as a long-lived species with complex brains and social structures, benefited more from caution than from dramatic renewal. The fossil record reinforces this story. Healed fractures, joint wear, and signs of past infection demonstrate that recovery often succeeded without perfect restoration. Individuals carried scars and structural changes for years, yet they survived long enough to contribute to their groups and pass on genes. Survival did not require flawless repair. It required sufficient function and stability. In modern contexts, this ancient compromise becomes easier to overlook. Medical interventions reduce infection and support nutrition. Surgical techniques align bones precisely. Yet the body's core behavior remains unchanged. It still rebuilds within strict boundaries. It still weighs growth against long-term safety. This perspective invites a shift in how we view slow healing. Rather than interpreting it as inefficiency, we can see it as evidence of restraint. The body does not pursue maximum speed because maximum speed carries hidden costs. It repairs steadily to avoid greater harm later. The scar that remains after injury is not simply a reminder of damage. It is a sign that stability was restored without risking chaos. There is also a broader lesson embedded in this pattern. Human survival has never depended solely on individual biological perfection. It has depended on cooperation, shared knowledge, and mutual support. Healing reflects this interconnectedness. The body does its part carefully, and the community fills gaps during vulnerability. When we ask why humans heal slowly compared to other animals, the answer does not lie in a single deficiency. It lies in a network of compromises shaped by longevity, complexity, and social life. Our biology favors durability over spectacle, caution over speed. As we move toward the closing reflections of this journey, it becomes clear that slow healing is not an isolated feature. It is woven into what it means to be human, long-lived, regulated, interdependent. The marks left behind by injury are quiet evidence of a strategy that values endurance above all. When we reach the end of this question, why humans heal so slowly compared to other animals, we find that the answer is not dramatic. It is not a single missing ability or a flaw in design. It is a layered decision written into the body over vast stretches of time. Our healing is slow because it is careful. It is careful because we are long-lived. It is careful because our cells are tightly regulated. It is careful because infection once threatened every open wound. It is careful because growth that moves too fast can become dangerous. From the savannas where early Homo first walked upright, to the small bands that shared food and guarded the injured, survival was never about flawless restoration. It was about staying alive long enough to continue. A wound did not need to disappear without trace. A bone did not need to return to perfect symmetry. The body only needed to regain strength sufficient for movement, work, and connection. That standard functional survival rather than perfection guided the evolution of our repair systems. Over time, other forces reinforced this cautious strategy. The human brain expanded, demanding stability and energy. Lifespans lengthened, increasing the cost of uncontrolled cell division. Social cooperation softened the consequences of temporary weakness, reducing the pressure for extreme regeneration. Infection risk shaped the sequence of repair. Cancer risk shaped the limits of proliferation. Energy scarcity shaped the pacing of growth. All of these forces converged into the tempo we recognize today. A cut closes in days, but a scar remains. A fracture stabilizes in weeks, but remodeling continues quietly for months. Neural injury adapts gradually rather than reversing instantly. The pace may feel slow when compared to animals that regrow lost structures, yet it reflects a life strategy built around endurance rather than rapid turnover. Even in modern hospitals surrounded by technology, this ancient rhythm persists. Medicine can reduce infection, stabilize damage, and provide nutrients. It can support the body's efforts, but it cannot transform the fundamental logic of human healing. That logic is cautious, regulated, and protective. There is a quiet lesson within this pattern. Human survival has never depended solely on biological speed. It has depended on balance between growth and restraint, between independence and cooperation, between repair and preservation. The scar is not a failure of the body. It is evidence that the body chose safety over spectacle. When we look at our slower healing through this wider lens, it becomes less a weakness and more a reflection of what we are, a species shaped for long lives, complex minds, and shared existence. We did not evolve to regenerate perfectly. We evolved to endure. And perhaps the deeper question is not why we heal slowly, but what kind of life required such careful healing in the first place. When we look back across this long journey, from the earliest wounds in prehistoric landscapes to the quiet scars we carry today, a gentle pattern remains. Human healing was never designed to impress. It was designed to protect. It does not rush because it cannot afford to rush. It does not rebuild perfectly because perfection carries risk. Instead, it restores enough, strengthens gradually, and preserves balance across time. Our species chose a different path from animals that regenerate dramatically. We invested in large brains, long childhoods, extended adulthood, and deep social bonds. These traits required stability. They required careful control of growth. They required systems that reduce long-term risk, even if that means slower visible repair. In that sense, every scar is a small record of evolutionary compromise. It marks the place where safety was prioritized over symmetry. The same is true for inflammation, for controlled cell division, for the cautious limits placed on regeneration. Each system reflects a calculation shaped by ancient conditions, limited energy, microbial danger, long lifespans, and interdependence. The body does not seek flawless restoration. It seeks continuity. There is a quiet comfort in this understanding. When healing feels slow, it is not because the body has failed. It is because it is following a rhythm refined over millions of years. Beneath the surface, cells are moving with discipline. Growth signals rise and fall in measured ways. Tissue strengthens in stages. The pace may test our patience, but it reflects restraint, not weakness. This perspective also invites patience with ourselves. A body shaped for endurance will not repair like a creature built for rapid turnover. Recovery requires time. Rest matters. Nutrition matters. Social support matters. Just as early humans healed within community, we still heal more effectively when stress is reduced and connection is present. Our biology remains intertwined with our environment and relationships. Modern medicine can assist and protect, but it does not replace the deeper design. We still carry a body adapted for ancient landscapes. Its priorities are stability, regulation, and long-term survival. When we honor those priorities through care, balance, and understanding, we align ourselves with the logic that sustained our ancestors. So the next time a wound takes longer than expected to close, or a scar remains after recovery, it may be seen differently, not as evidence of inadequacy, but as a reminder of the long evolutionary story that shaped us, a story in which speed was less important than survival, and perfection less valuable than endurance. Human healing is slow because human life was built to last. The body repairs carefully so that it may continue across years, relationships, and generations. In that quiet steadiness lies the deeper strength of our species. And perhaps the final reflection is this: if our biology chose stability over spectacle, what other quiet strengths might be hidden within the limits we sometimes mistake for weakness?